Hello and welcome, dear students. How are you all? So I welcome you to the class of biology. Dear students, please get settled. Dear students, please get settled quickly, and we are beginning right away. All right. Centers, please report your attendance. Yeah. Centers, please report your attendance. Shillong students, please get settled quickly. And I also sent you a PPT for the droppers. Yeah. So I have sent you a PowerPoint presentation for the cell chapter, and regarding this chapter, this PPT will help you in covering a few topics. And there is one more thing for the cell chapter. There are also a lot of fill in the blanks for your chapter. Okay. So when you study the PPT, please also solve the questions that are given in that PPT that I have made for you. So I'll be taking that shortly. Okay. All right. Good afternoon, Shillong students, and good afternoon, Itanagar. I believe the Chura Chandpur students are not yet online. So as they come, we'll be dealing with a lot of things. Okay. So in this picture here, this is a cell undergoing karyokinesis, which is basically a cell division. Okay. It is a part of cell division. Half a part of cell division is nuclear division, and then the cytokinesis is happening. So here, what we can clearly see are 4 nucleus formation. Okay, formation of 4 nucleus, and actually this is cell meiosis. And after this meiosis is covered, the thing is, it will neatly divide itself into 4 cells. Okay, so cytokinesis will happen after the meiosis has fully completed. 4 nucleus have separated. So there will be formation of 4 cells after this formation. Okay. So dear students, let's start from where we left yesterday. Okay. So I have something for you. Just a second. I have something for you. I have a cell division cycle. Okay. So first of all, I am going to show you cell division process as it is happening under the microscope. Okay. So please see this, and this will be helpful in enabling you to understand what's happening. Okay. So let's start. I believe you can hear this.
Orchestrated steps called mitosis. Every day our bodies must produce millions of skin cells to replace those lost through normal activity.
So this is
each of these cells must have a complete complement of the genetic material.
The cell growth, cell
prior to cell
process.
Identical cells, the DNA needs to be replicated so that each daughter cell receives an exact copy. Following DNA replication, the chromosomes condense in the nucleus of the cell.
This is the chromosome. Chromosome, I have shown the uncoiling here. Okay. So please see carefully. The chromosome is uncoiling itself into DNA plus histone proteins. Okay. So that is also clearly visible here. The chromosomes are condensing in the nucleus, and after they have fully condensed, the nuclear membrane will disappear. So chromosomes will come into cytoplasm, and as cell division is completed, it will again be decondensed into this chromatin material. Continuing.
Cores of histone proteins forming nucleosomes.
Here. It is called a nucleosome. Okay. And this is coiling and coiling, getting shorter and thicker. These are the cystochromatids. I have already told you about this. G1S, G2M, followed by cytokinesis. Okay. Can you identify this yellow colored cell organelle? Can you identify this yellow colored cell organelle here? I have told you about this cell organelle already. Just in the last class we have discussed about this cell organelle. What is the cell organelle? This yellow colored cell organelle that is appearing just below the mitochondria. What is the name of this cell organelle? Yeah, somebody can answer. Okay, you are passing your options to who will answer, right? Okay, I see what's happening in your class. Okay, nobody wants to answer. It's the centrioles, the centrosome. Okay, it's the centriole. It will form the spindle apparatus. Okay, lots of enzymes will be synthesized for the next phase, that is the M phase. Okay, so now the mitosis will happen, this yellow colored phase, and it consists of prophase, metaphase, anaphase, telophase, right? Prophase, metaphase, anaphase and telophase. So we have covered this. So please observe this. Look, I have told you in the last class that the central constricted portion is called the centromere. There is a button like structure on that, it is called the kinetochore. So this green colored structure is the kinetochore and the central portion is called the centromere. Alright. Continuing. Right. So now you can see how the spindle fibers are attaching to the central portion called the centromere. The movement phase. ऑडियो नहीं आ रहा था? हाँ, दे दीजिए. Okay, just wait for a bit. I'm actually did not play the audio. Just wait a second. So I'm going to play this from the start. Okay, so please observe the audio and
damage or rely on cell division. In eukaryotic cells, this process is accomplished by a series of well-orchestrated steps called mitosis. Every day our bodies must produce millions of skin cells to replace those lost through normal activity. Each of these cells must have a complete complement of the genetic material. Prior to cell division, where the cell divides into two identical cells, the DNA needs to be replicated so that each daughter cell receives an exact copy. Following DNA replication, the chromosomes condense in the nucleus of the cell. DNA condenses by wrapping around cores of histone proteins, forming nucleosomes. This beads-on-a-string structure is called chromatin. As a cell prepares to divide, chromatin coils up further, shortening and condensing the chromosome. The replicated chromosomes are called sister chromatids. The replication of DNA and the formation of sister chromatids is one part of the entire cell cycle. To prepare for cell division, the cell goes through interphase, which can be divided into three distinct phases. During G1 or gap1 phase, all the organelles and cytoplasmic components, including the centrioles in animal cells, replicate. Then during S or synthesis phase, the DNA replicates. Finally, during G2 or gap2 phase, all the enzymes needed to aid in the process of cell division are produced. Most eukaryotic cells spend a great deal of time in interphase, and a very short period of time actually dividing, a process called mitosis. The cell is now ready to go through mitosis, which consists of prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes condense and become visible, appearing as two sister chromatids held together at the centromere. The cytoskeleton disassembles as the spindle begins to form. In animal cells, centrioles play an important role in the distribution of the chromosomes in the dividing cell. The centrioles migrate to opposite poles, establishing a bridge of microtubules called the spindle apparatus, and the nuclear envelope breaks down. Towards the end of prophase, chromosomes attach by proteins in their centromeres called kinetochores to microtubules from each pole, moving the chromosomes toward the equator of the cell. During metaphase, all chromosomes are aligned at the equator of the cell, called the metaphase plate. Anaphase begins with the degradation of proteins that hold sister chromatids together, freeing individual chromosomes. The free chromosomes are then pulled by their kinetochores to opposite poles. At telophase, a cleavage furrow forms in the center of the cell. This indentation is made from a constricting belt of actin filaments surrounding the inside of the cell's circumference. Chromosomes cluster at opposite poles and begin decondensing as the nuclear envelope reforms around them. The spindle apparatus disassembles as the microtubules are broken down into tubular monomers that can be used to form the cytoskeleton of the daughter cells. In animal cells, cytokinesis completes cell division by extending the cleavage furrow to completely separate the newly formed daughter cells. Since plant cell walls cannot be constricted by actin fibers, vesicles form an expanding membrane partition called the cell plate. Like animal cells, plant cells use cytokinesis to finish the division of the contents of the cytoplasm between the two identical daughter cells. During the cell cycle, certain checkpoints are encountered to make sure the process is occurring accurately. And if it is not, the cell cycle will stop at the checkpoint and correct, or possibly inhibit that cell from dividing. The first checkpoint is the G1S checkpoint, and is considered the primary point at which the cell cycle continues or stops. External signals and growth factors can influence the cell cycle and affect the progress at or before this critical checkpoint. The G2M checkpoint allows cells that have successfully completed all three phases of interphase to begin mitosis. The last checkpoint is the spindle checkpoint, ensuring that all chromosomes have attached to the spindle in preparation for anaphase. Growth factors, the size of the cell, and the nutritional state of the cell are all contributing factors in cell cycle regulation, ensuring that only certain cells divide at appropriate times. Once all the checkpoints in interphase are cleared, mitosis can occur. From interphase to cytokinesis, the entire process of cell division can take on average 10 to 20 hours in a typical plant or animal cell. Depending on the nature and use of the cell, the process can happen at different frequencies as well. In humans, our skin cells have a high turnover rate due to wear and tear and go through mitosis very frequently, whereas other cells, such as adult neurons and muscle cells, rarely divide. The accuracy of mitosis, as well as the consistency of the checkpoints during interphase, ensure that most cells in a eukaryotic organism can produce identical copies of themselves. This process allows for growth and repair to prolong overall physiology as well as life itself.
Okay, so I have already told you that there are several phases of the cell cycle, and this is what we are doing right now today. So let's go through all this again once of all. All right. So here is what I was trying to tell you in the last class as well, that G0 phase is the cell specialization phase then when the cell is not dividing. As soon as it enters the interphase or the G1 phase, from interphase to G1 phase, so the cell is having some sort of preparation. Okay. Then there is the S phase, G1 after the G1 is the interphase S phase. Then there is a after S, G2 and M, after G1 S, G2. Finally, what we have is, please look at these notations. In interphase, the cell has 2n number of chromosomes and 2c concentration of chromatin material. Suppose it is 4 picogram, 8 picogram, it can be anything, any value. Okay. So 2c represents the amount of chromatin material that is present inside that nucleus. As it enters the S phase, the amount of chromatin material will be exactly doubled. So I am writing here 2c into 2 equal to 4c. 4c is the concentration of the chromatin material that is present now. Alright. So now the chromosomes, like single red thread has developed a double red thread. You can see here in this diagram here, everybody. Yeah, you can see in this figure here. I will also give this notes to you. Okay. So please see carefully here. Interphase S phase, concentration is 4c, but the number of chromosomes is still the same, 2n number of chromosomes still. All right. And after this is the G2 phase, 4c, 2n, no changes. But as the chromosomes have entered the prophase, metaphase, and finally the N phase, now the chromosomes have divided. Okay, the chromosomes have divided in anaphase. So now the concentration is 4c, but the number of chromosomes is 2n into 2, two sets of 2n. In telophase also 2c into 2, 2n into 2. But as the cytokinesis is completed, we are forming two cells completely. So 2c into 2, 2n into 2 forms two nucleus. Each has 2c, 2n chromatin material. Yeah, it is clearly visible here. So there should be no doubt regarding the amount of chromatin material. There should be no doubt regarding the number of chromosomes. When you see this figure, this is a very clear cut figure for everybody. This diagram should not leave any room for confusion for any kind of problems based on the number of chromosomes and the chromatin material concentration. I hope your doubt is clear now. Okay. So some people had asked me about this. So this is the answer for that part. Okay. For the concentration of chromatin material and the number of chromosomes. Okay. So mitosis we had completed yesterday. Now I had just given you a very quick revision of mitosis. Now I will show you what mitosis looks like under the actual microscope. So this process is very fast. This is a very fast process. So let's see how this is happening. Okay. So you can see this. Okay. So mitosis, meiosis. Just a second. Okay. So I have this video here. Mitosis, somatic cell division. Yeah. Okay. So I'm playing this for you. So this is how, if I see under the electron microscope, the light microscope, how the cell is actually appearing. So it's a very fast process. So please pay attention. It's a very fast process. There is no sound in it. It's just the image under the microscope. And it is happening very. It's appearing also very fast because it has been fast forwarded. Because this process over the process of 1 hour, it takes about 1 hour for this process to be completed, and this 1 hour process has been condensed into 30 seconds or 45 seconds. Okay. So right now what you can see here, no sound in it. Sound नहीं है इसमें. Sound है नहीं इसमें, जरूरत नहीं मुझे. Okay. So, in the beginning, please see here, the chromosomes were not formed fully. Chromosomes were not formed fully. Okay. In the beginning, the chromosomes were not formed fully. Now they have started condensing, shortening, thickening, and now you can see the chromosomes are quite well distinct as we are moving forward, right? So I am just forwarding it 5 seconds at a time. Now you can see at this point the chromosomes are very well distinguished, right? You can see everything like here. Now the chromosomes are arranging on the spindle apparatus. See this now. They will now arrange symmetrically. Okay. Yeah, you can, at this point the chromosomes have arranged symmetrically on the equatorial plate, and now they will move towards the opposite poles, right? So it's a really very beautiful process. It's in 3 dimensions. So what you were seeing now, the diagrams, they were just like seeing it in 2 dimensions. But now you can see how this is happening so fast. You can see, yeah, you can see this. Just see at this point, the chromosomes have arranged on the spindle apparatus, and see what will happen in a second. You can see the chromosomes migrating beautifully to the opposite poles, right? And now the anaphase is completed, telophase is completing, and telophase is completed. Now cytokinesis is happening at this point. See, 2 cells have formed. Really very beautiful process. I'll just rewind it. Up till now, this stage, this was prophase. Now the chromosomes have arranged on the metaphase plate. So this stage is the metaphase stage. Up till this process, okay, like up till 34 seconds, it was only prophase. Now chromosomes will migrate to opposite poles. So see, chromosomes are migrating to opposite poles, metaphase is completed. So this stage is now anaphase. Anaphase is completed because the chromosomes have reached the opposite pole. So now this stage is the telophase. And after telophase is completing, telophase is reverse prophase, 2 nucleus will be formed. Okay. Yeah, this is looking black and white because this is observed under a polarizing microscope. Okay. So there is a process in which we use black and white kind of light, okay, using the shadows to observe the chromosomes. Chromosomes have a slight bit of shadow because they are very thick. So it's called phase contrast microscopy. Okay. So although the diagrams is not as good as we see in the book images, but this is the actual thing happening under the cell. Okay. So finally, I suppose everybody has a good idea of how the processes of cell division have happened. So finally, we can move on, and now let's complete first of all the significance of mitosis. Please put the heading significance of mitosis. Significance of mitosis. Significance of mitosis. So the significance of mitosis will be discussed, and you can also see that because of the mitosis process, there will be 2 cells that have equal number of chromosomes. Okay. Right. So significance of mitosis. All right. So when we talk about the significance of mitosis, the prime significance is just a second. There is there is equal division, of two cells resulting in, there is equal division of two cells resulting in exactly equal number of chromosomes. So this is called equational division. Equational division. Okay. Equal to equational division. Okay. Okay. So the other thing is, the result of mitosis is production of cells which are exactly identical. The result of mitosis is that there are two cells which are exactly identical, Okay. So in asexual reproduction, in vegetative propagation, and whether you are forming a clone of any kind of organism, mitosis is the only process that is involved. When we want to make a clone, when we want to make an exact copy of that parent organism, then I am always going to use the mitosis process. Okay. So because of mitosis, there are two cells which are exactly identical, and this is because of the symmetrical division of the chromosomes. The chromatin material is synthesized symmetrically. There is no exchange, no variation. So there will be the future generation that is going to be produced by this process. It is going to be exactly similar to the parents. Okay. Therefore, in vegetative reproduction, in vegetative and asexual reproduction, no, it's not a new chapter, it's a part of cell division. I'm just telling you that asexual reproduction and vegetative propagation, that is because of mitosis. For example, you have a banana plant growing somewhere. A banana plant is producing a very small banana plant at its base. That plant will give the exact same fruit as the parent plant. Okay. Mangoes, grafting. By grafting, you get a new plant that is having the exact type of mangoes as the parent plant. Any fruit trees grown by grafting. So because that is a product of mitosis. Asexual reproduction and vegetative reproduction, we get individuals exactly similar to the parent. Individuals exactly similar to the parent. not audible. You are not able to hear my voice, Shillong? Is my voice audible now? Okay, okay, all right. Okay. All right. So let's do a few more things now. After this, we have completed all right. So I have already told you that small cells are more efficient than the larger cells. Okay. So the small cells, like the cells shown at the left hand side, on that side the cell is having a large nucleus compared to the cytoplasm. So when the nucleus and the cytoplasm volume is very well balanced, the cell is most efficient and it is capable of fast growth. But as the cell grows in size, it becomes less efficient and it undergoes aging also rapidly. So cell division is because the reason is of cell division is because the cells are becoming too large and it divides, producing two daughter cells. Each daughter cell is an exact replica of parent cell. Okay, before that happens, we have to replicate the DNA, and so each new cell will have the same exact genetic information as the parent cell. Don't need to write this. I can give you this PPT. You don't need to write this. Okay. So chromosomes I have already explained to you. Okay, thread-like structures. So the behavior of chromosomes, I have explained to you that the behavior of chromosomes on the beads on a string arrangement, it is clearly visible, and G1S, G2, M phase we have completed. Interphase is the phase between two cycles we have completed. Okay, mitosis. Okay, so now let's wrap the things up and finish this mitosis once and for all. Prophase, metaphase, anaphase and telophase. Okay, prophase, metaphase, anaphase and telophase. Okay, so prophase is the first phase. Okay, and it's also the most longest phase. It's also the longest phase. Metaphase is that phase in which the chromosomes arrange symmetrically on the spindle apparatus. So this will be clearly visible in the following slides that we will do after this. In anaphase, the chromosomes move towards the opposite poles. So the chromosomes undergo division and the spindle apparatus will carry chromosomes like this. This also we have completed. The division of chromosomes happens symmetrically, and as a result the spindle fibers are pulling these chromosomes to their side. They are grabbing these chromosomes because the chromosome has undergone division. Splitting of centromere has happened like this. So each chromosome, this will go to the opposite pole, this will go to the opposite pole, and these chromosomes are pulled together by a button like structure called the kinetochore. In anaphase, the chromosomes will reach the opposite poles, and in telophase, the process is completed and there is formation of two nucleus. There is formation of two nucleus at the end of telophase. After telophase is completed, the cells divide symmetrically by cytokinesis like this. and the length of mitosis can be few minutes and it can be days also, years also, but normally it takes days only. Prophase, the first and the longest phase of mitosis. You can see the events that are happening here. Chromosomes becomes visible, centrioles separate, move to the opposite sides of the cell. The chromosomes become attached to fibers in the spindle at the centromere. Okay, but this process takes a lot of time. Okay, and nucleolus disappears, nuclear envelope breaks down. So prophase is a collection of all these complicated steps. All these steps take a lot of time, and after this prophase is completed, finally we can attach the chromosomes to the spindle apparatus. Now where is the spindle apparatus in this figure? Where is the spindle apparatus in this figure? This is the spindle apparatus. Okay, this part is the spindle apparatus. This is the spindle apparatus, this region and this region here. Spindle apparatus is beginning to form, it has not formed. It is beginning to form, it has not formed completely. Okay. In metaphase, however, this spindle apparatus fibers have formed fully. Right? In metaphase here, the spindle apparatus fibers have formed fully. So it lasts only a few minutes. The chromosomes line up across the equator of the cell. The microtubules connect the centromere to the chromosome, and the centromere is attached by the help of spindle fiber to the pole of the spindle apparatus. So each chromosome has two kinetochores, so it will be attached simultaneously to the two poles, and it is hanging somewhere in between like an acrobat. Anaphase, what is happening in anaphase? The chromosomes are moving towards their respective poles because the centromere has been split into two pieces. Sister chromatids separate and move to the opposite poles. Anaphase is ended when the chromosomes stop movement. Only then anaphase is considered to be completed. Until and unless they are moving, at that time they will not be the completion of anaphase. So in early anaphase, the chromosomes have started moving. In late anaphase, the chromosomes will be stopping their movement altogether. All right. So you can see here, just a second. Okay. So this chromosome here, this one, okay, this chromosome here is moving towards this side, and when it, suppose it is reached here and it has completely stopped the movement. This is not a new chromosome, just trying to tell you what will happen after this has been completed. So at this point you can consider that the anaphase is ended. All the chromosomes will almost stop the movement at the same time. Next comes the telophase. In the telophase, the chromosomes begin to disperse or decondense into chromatin material. So if the chromosome is decondensing into the chromatin material, then that means that the chromosome is decondensing into chromatin fibers like this. It is decondensing into chromatin fibers like this. Nuclear envelope is reformed. So nuclear envelope is reformed. I have told you already that telophase is also called reverse prophase. Whatever happens in prophase, the reverse process happens in telophase. Okay. So the nuclear envelope reforms, spindle apparatus breaks down. So the spindle apparatus has broken down, so now it is again forming centriole, this thing here. The nucleolus also becomes visible. So nucleolus will also start forming here. At the end of telophase, the nucleolus will be completely formed. Cytokinesis, we get two cells, occurs at same time as telophase. cell membrane is drawn inward and the cytoplasm equally pinched into two parts. In plant cell, it takes place by a different process. I told you it is by the formation of phragmoplast, and the cell wall appears in the cell plate. Result is the formation of two identical cells. Alright, so now this is completed. Next, I have already told you what cyclin is. Cyclin is a protein which regulates the timing of the cell cycle in eukaryotic cells. Okay. So, the cell cycle, it is regulated by a protein called cyclin. This question has appeared in your NEET examination, that what is the protein which regulates the cell cycle? That what is the protein which regulates the cell cycle? This is very important for a NEET examination. Okay. Cyclin, this protein was previously mentioned in NCERT. 2-3 year old NCERT you might find that cyclin name is given. Now it has been removed, the question is still being asked. So this is very important for your NEET exam. I have already told you when I have given you the notes that mentioned the name of this protein. Internal regulators, they might also be responsible. Okay. So there are some proteins that respond to events inside the cell. So making sure that all chromosomes have replicated and all chromosomes have attached to the spindle apparatus before entering anaphase. So in the video I have already told you there are several checkpoints. There are checkpoints that decide whether the cell is ready for undergoing cell division or not. Okay. But what will happen if these checkpoints fail? What will happen if these checkpoints fail? What will happen? In that condition, cancer or cancerous cells will be the result of it. Okay. So cancer cells do not respond to these checkpoints or these signals that regulate the growth of most cells. Okay. And there is also gene. The name of this gene is called P53 gene. There is a gene called P53 gene. This gene halts the cell cycle until all chromosomes have properly replicated. But if this gene is damaged or becomes defective, then the cells can transform into what type of cell? The cancerous cell. Okay. So this is all about mitosis. This is all about mitosis. Now let's start meiosis. Please put the heading meiosis. Okay. Let's put the heading meiosis. Okay. Now let's put the heading meiosis. Cell division, meiosis. Okay. So what do you know about meiosis? In what kind of cells does meiosis takes place? My question to you is, meiosis takes place in fill in the blanks. Which kind of cells? I have already told you the answer. I just want you to remember it. In what kind of cells does meiosis happen? Do you remember? Students, do you remember? In what kind of cells does meiosis happen? Yes? You can at least try to answer. If you are wrong, I will correct you. I just want the name of the cell. If you are wrong, I will correct you. Okay, you have forgotten. Meiosis takes place in germ cells. Germ cells. In the germ cells, meiosis takes place. Meiosis takes place in the germ cells. Okay. So, can you name some germ cells in your body? Can you name some germ cells in your body? Sperm cells. Yeah. Sperm cells. Sperms and ova or egg cells. These cells in a body, they undergo meiosis, and now we must study the meiosis cell division. So, please listen carefully. Meiosis is divided into two phases. Meiosis is divided into two phases. Meiosis 1 and meiosis 2. Okay. Meiosis 1 is divided into karyokinesis 1 and cytokinesis 1. Karyokinesis 1 and cytokinesis 1. Meiosis 2 is divided into karyokinesis 2 and cytokinesis 2. Cytokinesis 2. Karyokinesis 1 is divided into prophase 1, metaphase 1, anaphase 1 and telophase 1. Prophase 1, metaphase 1, anaphase 1, telophase 1. Karyokinesis 2 is divided similarly into prophase 2, metaphase 2, anaphase 2 and telophase 2. Okay, prophase 1, metaphase 1, anaphase 1, telophase 1, prophase 2, metaphase 2, anaphase 2, telophase 2. All right. Okay. All right. So, now let us study these two meiosis, or we should study meiosis 1 first of all. So, please put the heading meiosis 1. Meiosis 1. Okay, meiosis 1. So, what happens in meiosis 1? In meiosis, I have few things for you planned out. Meiosis 1, prophase 1, metaphase 1, anaphase 1, telophase 1. So, let us start with the first. This is actually the telophase stage, meiosis 1. Yeah, this is the thing I wanted to show you all. This is a cell undergoing meiosis. This is a cell undergoing meiosis. So, on the left hand side, 10 micrometer is a scale of how small these cells are. Okay. So, these are 4 different cells. Okay. In the first cell, on the lower left corner, the meiosis has been completed. The meiosis has been completed. In the second cell on the top, the two cells on the top, they are undergoing meiosis 1. Okay. And in this lower cell here, this cell, this one cell, this is undergoing meiosis 1, and specifically prophase 1 is happening here. Prophase 1. Okay. The first stage of meiosis 1 is the prophase 1 phase. Okay. So, first of all, we need to study what is happening in the prophase phase of cell cycle. So, please put the heading that prophase 1, meiosis 1 is divided into meiosis 1 is divided into prophase 1, metaphase 1, anaphase 1 and telophase 1. Prophase 1, metaphase 1, anaphase 1 and telophase 1. Okay. But the prophase 1 of meiosis is very, very complicated. The prophase 1 cycle, the prophase 1 is very complicated and long, therefore, it has been subdivided into leptotene, zygotene, pachytene, diplotene and diakinesis phases. Okay. So, these are the phases that we are going to study. First of all, leptotene, zygotene, then pachytene, then diplotene and then diakinesis. Okay. Finish writing, please. All right. So, let us start here. Put the heading leptotene stage. What is happening in leptotene substage? So, let me give you a quick idea what is happening here. Prophase 1 is a long stage. In mitosis, prophase 1 meant then when prophase is starting, at that time nuclear membrane starts to disappear. When prophase is ended, the nuclear membrane has completely disappeared. Same is the thing with these 5 substages. When leptotene is starting, nuclear membrane starts to disappear, and when I complete the last phase, which is the diakinesis, nuclear membrane will have completely disappeared at all, and chromosomes are undergoing shortening and thickening from lactotene to zygotene to pachytene, diplotene and diakinesis. So at the end of diakinesis, we get a very short, thick and easily visible chromosome. All right. So we will get this. So let's complete this now. I want to show you something here. Anaphase 1, prophase 1, metaphase 1, anaphase 1, telophase 1, and so this should be sufficient for you. Just a second. Yeah, okay. Just a second. Okay, so here we have some chromosomes now. All right. So beginning from the start, so let's start now. Meiosis. It is a type of nuclear division, and it's also called a reduction division because it results in the production of daughter cells with half the set of original number of chromosomes. Okay, so we can say the daughter cells will have only one set of chromosomes compared to the parent. Okay. All eukaryotic cells, mostly they have diploid number of chromosomes. That means they have two sets of chromosomes. In mitosis, we always get diploid daughter cells, so they always will have two sets of chromosomes. But in case of meiosis, we will have only one set of chromosomes for each gamete. So when the gametes will fuse, then the embryo, then the zygote that is produced, first zygote then embryo, it will have two sets of chromosomes because both the male and the female sets have fused together. Each is bringing one set of chromosomes, the egg and the sperm. When they will fuse, they will form the zygote, and the zygote will have two sets of chromosomes. So chromosome number remains conserved in a species. It helps in preserving the karyotype or the number and essential physical properties of chromosomes. Okay. And all of our cells are diploid. Okay. So there are some differences between meiosis and mitosis. Meiosis has one round of duplicate of DNA, 2 rounds of nuclear division. But in mitosis, 1 round DNA duplication, 1 round nuclear division. In meiosis, homologous chromosomes are paired. In mitosis, this does not happen. Homologous chromosomes exchange genetic material in meiosis, particularly in prophase in pachytene stage. Okay, and this crossing over results in recombination, that is formation of unique chromosomes, new characters, and different from both mother and father. If this was not done, then all of you brothers and sisters would have the same character. Okay, so mitosis is very simple. Okay, chromosomes separate very neatly to the opposite poles. So diploid parent cell, chromosome duplication, metaphase, anaphase, telophase. Okay, it's very easy. But in meiosis, we have prophase 1, crossing over. Metaphase is joining at the equatorial plate, and then the separation. So meiosis is having a very key event called the recombination process, and this process is what we have to study right away. Okay. So, meiosis 1, right. So, let us go back a bit and we will continue this because this is a very important phase. It is going to come in the Pachytene stage. So, let us write a few bit of notes first and then we will come back to this. All right. So, please write the changes that are happening in leptothene stage. In leptothene stage, it is the first stage of prophase 1. It is the first stage of prophase 1. First stage of prophase 1. It involves condensation of the chromosomes. Now, the chromosomes have undergone DNA duplication. So, both the all the chromosomes will have 2 sister chromatids. Okay. All right. So, leptothene stage, leptothene is very short. Leptothene stage is very short. Leptothene stage is very short. Okay. Also, the chromosomes, the sister chromatids are very close together. The sister chromatids are so close together, that they appear to be like a single thread, of single chromatid, because they are very close together. If they were separate, then you could have easily distinguished the 2 chromosome sister chromatids. Okay. All right. So, let us draw the leptothene stage. In the leptothene stage, you can see here, you will have to draw a large, sufficiently visible structure, and in that I am going to label a nuclear envelope that is disintegrating. Now I will rub some portions of it. Like this. Let us draw 2 chromosomes here. Let us draw one with the red color and the other with blue color. chromosomes are long and slender like this. So we will draw together 1, 2, 3, 6 chromosomes. Let's draw 6 chromosomes. So let's take another one of blue color. Like this. So this will be the first homologous chromosome that we will discuss later. Okay. Or maybe let's take only 4 chromosomes, because that way it will be easier for us to understand, because the space will become quite less. So although they look like single chromosomes only or single chromatids only, actually they are having double sister chromatids. They are having double sister chromatids. So these are homologous chromosomes. Homologous chromosomes. Okay. And they are long, thin and slender. They are long, thin and slender. Okay. They are long, thin and slender. Alright, let's move on to the next stage, the zygotine stage. I hope you have drawn this figure. Should I move to the next? Okay, I hope you have drawn it. Okay, I believe as you are drawing also, I can give you a 5-minute break. So, because you are taking some time in diagrams also. So, take a quick 5-minute break, and after that we will return. I will show you the other things in this regard. Okay. So, draw the diagram and go for your break, and return after 5 minutes. Okay. Thank you. Okay, so now we begin the zygotine stage. The zygotine stage of meiosis. Now please understand that the chromosomes that have started to condense, now they will be actually paired together. So the chromosomes that have been resting up till now, they will now form a complex called the sandwich complex, and this sandwich complex will consist of a protein network which will help in joining this chromosome to the next chromosome, and this chromosome complex, by the process of pairing, it is called synapsis. So please write, in this substage, synapsis, a very, very important process. Synapsis occurs between homologous chromosomes. Synapsis will happen. And what is exactly happening in synapsis? This process is happening. So please look carefully. There are two chromosomes. One is shown in red, one is shown in blue color. The sister chromatids are basically undergoing an overlapping. So the first diagram here, it represents the synapsis between the chromosomes. In the next stage, in Pachytene stage, the crossing over will happen, and this green and black colored complex, that represents the synaptonemal complex. Okay. So please put in your notes. Yeah. So please note, it is also called zygonema stage. It is also known as zygotine stage. It is also known as zygonema stage. And the synapsis can form up and down the chromosome allowing numerous points of contact called synaptonemal complex. Wait a second. Allowing numerous points of contact. The two chromosomes the two chromosomes are held together in position, the two homologous chromosomes, homologous chromosomes held together in place by the help of a protein complex called synaptonemal complex. Synaptonemal complex. Okay. All right. Moving on. Let's draw what is happening in synaptonemal complex stage or in the zygotine stage. The two chromosomes which we are calling homologous chromosomes, they are going to be held together in the place and just a second. Actually, there are three lines. We are going to draw three parallel lines, and these three parallel lines basically represent the synaptonemal complex. Or you can draw just two lines, and I'll put a filling substance in between, like this. So just like an Oreo biscuit, the sandwich complex is there, and this is called the synaptonemal complex. Synaptonemal complex. These are the non-sister chromatids of two homologous chromosomes. Of two homologous chromosomes. So when I talk about the second stage of meiosis, I am trying to refer to this synapsis stage. Okay. Synapsis or overlapping or pairing of homologous chromosomes occurs in which stage? Zygonema stage or zygotine stage. Have you finished drawing? Okay. All right. this has been done. Okay. So now let's this is the main stage. So if I draw the figure of the cell in which this process is happening, so the nuclear complex is dissolving and all that, the nuclear membrane is dissolving, and in this cell the two homologous chromosomes, they are undergoing pairing. So if I want to show you at the other stage, like this, okay. So this is how it is actually looking like. We have started with 4 chromosomes. So this is how the pairing is happening. Each homologous pair of chromosomes, it undergoes pairing in this way. Okay. And there is a sandwich complex or synaptonemal complex in between these chromosomes, like this. The blue colored, I have shown this by blue color, pink color. You can also choose some different color for representing it. So this is the basically the sandwich complex, this synaptonemal complex between 2 homologous chromosomes. So this is the zygotine stage. The zygotine stage. Okay. I hope. Have you finished? Let me know. Okay. Okay. So in this zygotine stage, the homologous chromosomes have undergone pairing. Now comes the most important stage on which many questions are asked, the favorite stage of your examiners of NEET exam, the Pachytene stage. The Pachytene stage. Most questions are asked from this stage only. Pachytene stage is most important. Okay. This is your most important stage, because in this stage the crossing over will take place. In this stage the crossing over will take place. All right. So please write now. Pachytene stage. One more thing. In zygotine stage, in this diagram, there is one more thing I need to tell you. This is called bivalent or tetrad stage of chromosome. This is the tetrad or bivalent stage of chromosome. Bivalent because 2 chromosomes are there, and tetrad because total 4 chromatids are there, 2 sister and 2 non-sister. Sorry, total actually 2 sister, 2 non-sister, basically because they belong to different chromosomes. So just write this in between. Just write this in between. Okay. All right. So please write packetine. Okay. In the packetine stage, this is the most important stage of prophase 1. This is the most important stage of prophase 1. Why? Crossing over takes place at this stage. Crossing over takes place in packetine stage. So let us understand what is crossing over. Crossing over is the breaking of two chromosomes at some specific points and exchanging exactly the same amount of DNA, exactly the same amount of chromatin material with the other pair of chromosome. Okay? Crossing over is the exchange of genetic segments, chromatid segments, chromosome segments with the other pair of that homologous chromosome pair. There are two members, right? So between two non-sister chromatids, exchange will happen. So let's see this exchange in person, how this change is happening. Centromeres are overlapping with each other. First figure, the synaptonemal complex has formed. Second figure, in the third figure, you can see the non-sister chromatids are undergoing breaking, and after breaking, there is reunion, there is rejoining, and this rejoining is shown in the last diagram here. So the blue part is exchanged with the pink part. It is visible to everybody, right? So please draw this diagram now. Please draw this diagram. You have to draw this diagram. Just a second. So this is the synaptonemal complex, right? This is the first homologous chromosome. This is the second homologous chromosome. Okay. So they both belong to the same pair, of course, right? So they belong to the same portion. Just a second. You can also call it homologue, it's fine. You can call it homologue, there will be no confusion. Okay. And these two non-sister chromatids are undergoing breaking. Okay. Breaking and reunion. So this is called breaking and this represents reunion. Crossing over is explained by breakage and reunion theory. Breaking and reunion by two scientists, Stern and Hotta. Just draw this diagram, please. First of all, please draw this diagram. This is happening in packetine stage. This is happening in packetine stage of prophase 1. You can also write the notes if you want to, but the diagram is more important. The diagram is more important. Let me know as soon as you have finished. Okay. Please finish it fast. Have you done it? Okay, yeah, the time is also coming for the class to get over. So, okay, I think I'll, this is your last part that I will do today. In the next class, we will continue with our present chapter, that is meiosis. And after meiosis, I will be taking your next chapter, okay. So, meiosis takes a bit time, and after that we will continue with the next chapter, okay. So, finish this slide and then we will, then you can all go home, okay. And please also complete the homework that I have given you. I have given you a PowerPoint presentation. In that there are some fill in the blanks questions. So, just you have to just read that, and there are a lot of diagrams in it. So, you will not have any problem for that. Just read that, solve that, fill in the blanks by yourself, okay. And if you have any doubts, please come back to me, okay. So, that will help you in completing it, and I will send you this PowerPoint presentation to you as well, okay, and then I will send you the notes as well. Thank you, dear students. I will meet you in the next class, and in the next class we will finish meiosis, and we will take a look at the video for the meiosis, and this chapter will be over. We will solve a few questions also. So, thank you. I will take your leave now. Go home and have safe. Yeah, some people are celebrating holidays also, okay. So, good luck. Thank you. See you in the next class.